Good morning and welcome to our second day of Big Data and AI Forum. The activities, we will start with the first panel for today devoted on digital health, and later it will follow about future cities and disinformation we are working on. So the activities in digital health domain at GATE are in developing and deploying artificial intelligence, machine learning, biostatistics and bioinformatics methods to biomedical problems, especially those related to cognitive diseases. And our main focus is Alzheimer's disease that affects millions of people in the world. Its causes are mostly unknown, and currently it's not possible to reverse the cognitive decline, although a new medication was recently approved. And different types of data are typically collected during the Alzheimer's disease studies: phenotype data, genetic data, imaging, there are a lot of data, and gaining insight from this available data requires analytical skills and competencies in many fields, and this is not possible to be done by one team only. Because of that, at GATE we're working together with our partners and colleagues from leading universities like Stanford, Oxford, and Chalmers. And now in this session, you'll see some of these collaborations. And being a big data institute, GATE in digital health domain, we are working really on the large data sets. For example, for some of our projects, we plan to process more than 5,000 terabytes of 500 terabytes of genetic data from Alzheimer's disease patients and healthy controls. So we also simulated many thousands of databases of synthetic data and imputed data. And in this panel today, our lead in digital health domain in GATE, Diane Palejev, will describe some of our projects and in more detail and together with our partners. So with this, I will give the floor to Diane to moderate this panel.
Thank you, Professor Ilieva. Indeed, the Alzheimer's disease poses various problems that require different points of view and different competences. And for that reason, today we have colleagues with various backgrounds and competences that will talk about different technologies, methodologies, and approaches to various problems that are related to the diseases. First, we have our keynote speaker, Alex Urban, who is an associate professor in psychiatry and behavioral sciences and in genetics at Stanford. And of course, for that reason, given our interest in cognitive diseases, it is not a surprise that we are collaborating with him and his group. And of course, his talk would be on genetics, namely complex structural variants in the human genome. Alex, please.
Fantastic, thank you. Thanks for the introduction and the invitation to participate in this event. Thank you very much. I don't see my slides yet. Are they up, and I just don't see them, or? Ah, there they are. Very good. Let me quickly try let's see. Yep, I can. No, yeah, very good. Good. And I'll start my timer here. Yes, I want to talk about complex SVs, complex structural variants. But before I do that, I want to spend just a few slides kind of framing the background a little bit. And since I have a tendency to meander a bit, I threw a couple of bullet points here to achieve that framing. And my point is that while on the one hand, standard whole genome sequencing analysis of human genomes is now readily available and being carried out in large numbers, and also moving in the clinic in the context of digital health, but standard whole genome sequencing defined as short reads at 30 to 40x genome coverage, and then standard analysis pipelines will actually miss many types of sequence variants, for example, in repeat regions, haplotypes, and the complex SVs. And even long read whole genome sequencing can only partially remedy this. And but the good news is, and in that context, I would then want to also talk about SVs, is that we can use, we can develop advanced approaches, either experimental, computational, or both, that can then help us and allow us to reveal types of genome variation that is currently invisible when doing standard whole genome sequencing analysis. Let's see, next slide here. So I don't have a pointer, but looking at this, breakdown of the human genome sequence, you see in the lower right, the 2% of protein coding genes. And then basically the whole, more than half, when you look at where it says lines, the red part, all the way over to segmental duplications, 5%. That part is inaccessible to standard whole genome sequencing to date. And I want to show very briefly mention two other projects that we recently completed, where one can remedy this by developing advanced either computational or experimental or combined approaches. And that's a bit self-serving that I showed these two things from our lab, but I know them well, and I think they serve well to in the context of this framing purpose. So one, you see here somatic line 1 insertions in human neurons and glia. So again, going back one slide, you see line 1s make up to 20% of the human genome. And you will actually get reads, if you look in panel A here, that span both the unique sequence and line 1 insertions, either existing ones or novel insertions. But the standard pipelines will throw that out. Well, there are pipelines that then look for that, that you can add on already in to find germline insertions. But it gets really tricky because these line 1 transposons also sometimes jump in somatic tissue, so they are present then only in a fraction of the cells from which you extract your DNA. And so we developed this machine learning based approach. We use this pedigree data on the right here to train the model. I don't have time to go over any of the details, but the bottom line is at the bottom when we do that, and we did extensive quality control of this because it's an extremely noisy problem. That we can now confidently detect line 1 insertions in somatic genome sequences, even if they're present in only as little as 1% of the cells from which we sample the DNA. And we've been applying that to brains, to DNA extracted from autopsy brains across a variety of neurodevelopmental and neuropsychiatric conditions. And we find, well, we confirm what was already known, that yes, these line 1 insertions occur somatically in human brains, but they actually occur because you can trace them across both hemispheres. They are present in both glia and neurons, and they are present at relatively small, lower frequencies, we find now, below 1% to up to 14%, but they insert often in very interesting functional regions of the human genome. And I don't have time, that's an entire separate talk. But so that's one example for advanced analysis methods of whole genome sequencing data. I'm trying next slide. Oh, here we go. Let me see, one back again, and then here we go. Yeah, very good. And then the other example on the lower right, this bioarchive manuscript, where we developed something called CTLR-seq, which is basically a combination of CRISPR-Catch and long read sequencing and short read sequencing. And we used that, we developed that to address the problem of analyzing segmental duplications, which you see kind of in the upper left here. 5% of the human genome, and specifically the human genome, are made up of these segmental duplications. They are very interesting in the context of structural variation analysis, and they're effectively impenetrable to genomic analysis. You see here whole genome sequencing data from 6 people, 3 controls, and 3 patients with a very large deletion on chromosome 22. And you see you get fairly clean signal outside of the sec dupes. You see in red these boxes at the bottom of the graph, sec dupes. Right, and outside you can see where there's 2 copy numbers and 1 copy numbers in the data. But where the sec dupes are, you get basically just these firestorms of signal, and you can't determine anything. And so, for example, we can't tell when we look at these 3 patients, where actually do their deletions start and end? And this is an uncertainty of up to 800,000 base pairs between these 3 patients. And so so what we did here is we with this scheme where we do CRISPR as cutting of DNA that has been extracted from cells, is immobilized in an electrophoresis gel, and then we cut out the SEC dupe regions that we want to look at. We run the pulse field gel, we switch the current, we extract the DNA, only that part of the genome that we want. We basically throw away the rest of the genome and we extract these 400,000 base pairs where the deletion happened, and that is SEC dupe sequence. And then we sequence that with long reads, assemble, and pile short reads on top. And then we can resolve these regions now that were previously inaccessible to analysis. So so now on to the complex SVs. So now thinking of the other almost half of the genome, right? That starts with protein coding genes and then going clockwise up to miscellaneous heterochromatin. So these non-repetitive parts of the human genome, for those we have fairly good, when you do whole genome sequencing, short read whole genome sequencing, we have very good, actually very robust analysis type of point mutations and small indels, and also for non-complex SVs. So you see on the left there, you have translocations and deletions and duplications and insertions. And we can call these fairly well now, no, quite well actually, as long as they're simple SVs, meaning they have only 2 breakpoints. They have a start and an endpoint, and then something happened to a long stretch of DNA sequence in between. And these are often very disease relevant. But we've known for years that complex SVs are present in the human genome. We just couldn't do much about them. And so a complex SV is defined as an SV that has more than 2 breakpoints. It's at least 2, but often more distinct SV signatures in a single mutational event. And what you see here are 3 examples of actual complex SVs that ARC-SV, the algorithm that I want to present a bit about, did pull out of 2 genomes that we had. You see the publications at the bottom there, had studied in great detail with it. For example, on top the example you see there, the segment C has been inverted and B and D have been deleted, right? But then it also can get really complicated more to the bottom. So this is not a trivial task to achieve with the existing algorithms for reasons that I don't have time to explain. However, this is a timely topic. People are now turning towards this in the field of genome sequence analysis. There was this recent paper here where they analyzed 14,000 genomes, mostly European and some African ancestries, and they found more than 5,000 complex SVs. Shown, depicted on the right. And the way they did this basically on foot, the hard way. They used existing algorithms, looked for irregularities in the signals, and then sorted things out by hand. And so this is where ARC-SV comes in, which is an algorithm. It stands for Automated Reconstruction of Complex Structural Variants. And it was developed by a graduate student here at Stanford in Bing Wong's lab in statistics, and with whom we are collaborating on many genomics projects. So what it does, it collects under A, it collects from standard whole genome sequencing data. It collects clusters of split reads and clipped reads and discordant paired ends. And then under B, it does something called constructing an adjacency graph, which I don't have the mathematical skills to understand, but the people who do in our lab and amongst the collaborators tell me that this is a good thing. And then it proposes candidate SVs based on those matching adjacencies. It evaluates diploid genotypes against observed alignments, and then comes up with the most likely a complex SV, and it actually makes nucleotide level predictions. And I'm an experimentalist. When I first saw this, I didn't quite. It seemed too good to be true. So we did extensive quality control that I'm completely leaving out otherwise of this talk. But we did dozens and dozens of long range PCR and Sanger sequencing and a variety of other quality control measures. And this thing really works. We achieve. We achieve validation rates based on its predictions of something like 85% or so. So what I'm going to do now is, the main algorithm has been kind of lingering on bioarchive. So we teamed up with Impalayev to kind of produce a nice large use case to get this algorithm out and then start using this algorithm. So what we've done now is, in collaboration with Impalayev, we've analyzed more than 4,000 whole genomes, standard whole genomes, from across the world. And what I'm going to show you is where we are right now, with sifting through these results. So actually looking only at a subset at 2,500 of these genomes here, it is a PCA looking at the population and then colored by the populations that these genomes come from. And you see here a distribution that makes a lot of sense. You have Africa in red on the side, and then you have the other populations. So this is more quality control. This is how this should have looked. Same when you go to a higher allele frequency. This is something that I didn't see coming. When you plot these complex SVs in their genomic position, even though we, of course, blocked out telomeres and centromeres, we see that on many chromosomes, we see these clusters of occurrence near the ends of the chromosomes. Here's some larger chromosomes, with smaller chromosomes. We don't quite know what that means. I'm speculating that it has maybe something to do with the mechanism of formation of these complex SVs, but that'll be something that we'll have to look into some more. You see, there are some predominant classes of these complex SVs, and then there's also a whole string, a large group of others that can't be easily classed. The size distribution, ArcSV has a soft upper size limit of 100,000 base pairs across the different classes here. You see they're more small to medium size, 1,000 to 10,000 base pairs. There's a lot happening. And then the last few slides, in the interest of time also, we assigned then to have a first look at the functional relevance potential functional relevance of these complex SVs. We assigned the nearest gene for each of these SVs, complex SVs. And then, for example, here we run a pathway analysis, and to our surprise, but pleasant, happy surprise, since we're also in the Department of Psychiatry and we're interested in brain development, brain function, there's a lot. I mean, all the top pathways and functional categories that come up have to do with brain development and brain function. Here's another way of looking at it, where we took these complex SV associated genes and compared them against gene expression out of the GTEx project in different human tissues. And again, you see in the top left, all these bars, there is a lot of upregulated genes associated with complex SVs that's almost all in various brain regions. And I should mention going back 2 slides. ArcSV does also call simple SVs, with 2 breakpoints, which I left out here. But when we do that same thing, assigning the nearest gene and then looking for functional categories, there is no such enrichment for brain functional categories. So that's very interesting. And then my last slide is here we tried an association, use that term loosely, of the complex SVs with marker SNPs that came out or groups of marker SNPs coming out of multiple GWAS, genome-wide association studies. And you see here there's kind of a wide variety of phenotypes that these studies were looking at. You see height, morning person, photic sneeze reflex. But there are also, again, quite a few, there's a lot of disease-relevant phenotypes there, and again, brain-relevant phenotypes, right? There is schizophrenia, and since in particular, since also it was mentioned, and we're going to keep working with Dean and he has a big data set lined up of Alzheimer, even in the control. Now this is all based on healthy control subjects, right? But even there, the positional association at least is for Alzheimer's disease. You see they're kind of towards the bottom of the list. And so with that, I will end here. And say my acknowledgements and thanks. So in my lab, Bo Zhu and Chris Hughes were the driving force behind this, with some help from Renal and Jawei. And then, as mentioned earlier, the algorithm was developed by Joey Arthur when he was in Wing Wong's lab and under the guidance of Wing Wong here at Stanford. And we're now working in expanding the use while refining its quality of this approach. Working with Dean Paleyev at the Gate Institute. So thank you very much again, and happy to take question or discuss. Okay.
Thank you, Alex. If I could just follow up a little bit, what you mentioned about applying the algorithm on Alzheimer's data. These are the 100 terabytes of data that we have from sequencing of Alzheimer's disease patients. And a related biological question. Are there any hypotheses or proven facts that more complex diseases that manifest in different ways might be somehow related to more complex variants in the human genome?
Yeah, interesting. Well, we know that in terms of complexity, for example, for something like Alzheimer, for the more common forms of Alzheimer, right? There are some early onset, clearly familiar types of Alzheimer where, you know, there are some strong candidate genes, individual candidate genes, individual smaller mutations. But for the, you know, for the more common type of Alzheimer, we know it's highly genetically, there's a strong genetic component, but we know that the genetics has to be complex, but more in the sense at this point of it has to be very likely more than one locus involved in a person, right? And that actually came out of, for example, some, especially recently, several very large GWAS analysis that come up with, I think we're at 200 or so SNP-based markers, SNP-based loci for Alzheimer, right? And they're spread out all over the genome, and they're actually individually common in the population, these markers. So everybody probably has one or the other of them. So one way of thinking about this is that, well, it's maybe the disease risk and the disease predisposition comes from having inherited a particularly bad, speaking sloppily here, a particularly high risk combination of these otherwise common loci. And so that there's one level of complexity. As to what then, now these are marker SNPs. Most of them, if not all of them, but most of them will not actually have a function in and of themselves, right? They're just tagging some genetic variant more or less close by. And as to the nature of that, we don't know. It could be anything. It could be a point mutation, or it could be a complex SV somewhere within you know the association range. Is it more likely that it's a complex variant? Let me think about that for a moment. I mean, it could really be a mix. I wouldn't want to put my eggs in one basket there. It could really be a mix, but it is, yeah. So that's where we are, I think, with this.
So I guess we would need to kind of in the downstream analysis deploy different types of methods, bioinformatical, but also machine learning methods that really catch nicely, let's say, interactions between different features, or in this case, events.
Yeah, how to go about this? This will be keeping us busy for the next few years. Sorry, I cut you off.
Yeah, indeed. And also you mentioned that the previous effort to find complex SVs were done kind of manually or semi-manually. So from your point of view, of course, your expertise is in genetics, biology, and so on, not in machine learning, but how did the machine learning approach help in solving that problem? Because obviously, especially now we are looking to do this on a large scale for many patients, and it's perhaps infeasible to do that manually. So how do you see the role of the machine learning
so that's
doing that automatically?
That's right. I mean, the advantage of doing this automatically is, well, one is obviously, you know, speed and throughput, and not abusing people by, you know, making them perform the really dull task of sifting through all these tentative signals. There's also, it seems, so we're doing a lot of benchmarking right now still of ArcSV relative to the other approach, not actually necessarily with the GNOMAD paper, but there's something else on bioRxiv where another group analyzed the same 2,500 thousand genome sequences using a similar kind of on foot or same image. And so we're benchmarking against that also. And so far it looks like ArcSV vastly, like almost twofold outperforms the semi-manual calls in terms of accuracy, specificity. So there's not just speed and in throughput, but also it seems just accuracy, quality of the calls. And so and to the extent in which machine learning, I'm the experimentalist, right? So I'm only learning very slowly so it's human learning on my part as to what machine learning even is. I'm being told that ArcSV itself, at its core, does not use machine learning, but we've been using machine learning to clean up the output, right? So yes. I left all of that out of the talk, right? But so we've benchmarked. We have some very high quality, long read, assembled, high confidence genomes that we use at the ground truth, and then use those to train a model that then helps us sifting through what an initial ArcSV run spits out, produces, and then that's then our final call set. So machine learning comes into play kind of after ArcSV has done its initial graph-based calling.
So it's a combination between, let's say, bioinformatical approach and machine learning substituting for the DOE or routine work
that's right, exactly.
Otherwise done manually. And this is actually a pretty good use case for machine learning that in many cases is unnecessarily used, but in this case, as you mentioned, it performs much better than even the manual or semi-manual approach that exists.
It really very impressive. And also, sorry, just mention machine learning one more time. I'm really a true believer now. I had of it before we used it for the first time in my lab, and I had no real idea what it was until we applied it to this problem that I mentioned very briefly early on to detect line 1 insertions in the genome. And this was really what I briefly showed was really the result of 8 years of work, and the first 4 years or so we tried without machine learning, and we got nowhere. I mean, it was all dead ends. The noise was so overwhelming. And then we figured out, we turned to machine learning and we trained a model. And then all of a sudden we started getting good calls that we can go back then experimentally to the DNA, and the line 1 insertion has happened exactly where the model said that it happened. So I'm really impressed by how powerful this was and how it just and the
moral of the story is go ahead.
We also there we did some benchmarking against the existing algorithm, which is not bad. There's one algorithm that does something similar, looking for somatic line 1 insertions, but it really has confidence only. It makes confident calls only if they're present in something like 25% of the cells at the point where the DNA is sampled. And again, our approach now makes very clean calls, even if a line 1 insertion is present only in 1 out of 100 cells, 1% of the cells where we sampled. So it's also much more sensitive than But sorry, go ahead. I cut you off again.
Yeah, let's conclude with the fact that this is a really good case for applying machine learning methods when they are really needed, and if that is the case, they yield really good results. And of course, we still have the task of applying RSV to the genetic data from many patients from Alzheimer's disease. That's going to keep us busy for several months.
That's right. Looking forward.
Okay, thanks. Thank you
Fantastic.
So much. I know that is
thanks again for having me.
Way past midnight over there. So thanks a lot.
Yeah, I'm sorry. Usually I would stay on, but I have to
presentation.
Great. Thanks again. Thanks. This is great.
Thanks. Okay, so let me mention a couple of things that we do at Gate. If you could pull my presentation. Next slide. Yes. So very briefly, we do many things here at Gate related to Alzheimer's disease. Next slide, please. Our flagship project is what we call omics type of analytics. This is basically involving all sorts of different types of data and creating models from data that is not necessarily types of data that is not necessarily comparable between each. Also, and you're going to see this later, we are doing risk calculator for pre-screening, and that risk calculator estimates the risk of Alzheimer's disease. The goal of this calculator would be to avoid expensive and potentially really intrusive procedures that could be applied to potential patients. In general, and a good example of that was Alex Urban's presentation. We do a lot of analysis of next generation sequencing data. And to some extent, we cover some border or edge cases, because ideally one would have lots of data, but that is not necessarily the case. Especially with clinical trials, there is often missing data. And for that, we work on missing data imputation, because we don't want to necessarily throw out observations or patients for which some of the data is missing. Also, in many cases, we do not have the necessary amount of data so that we can draw meaningful conclusions, and for that we use simulated data, and this is one of our main topics of work. I should say we work on information systems that are related to the digital health domain. For example, the Cognition project and some pilot projects that we have done. For example, on single-cell sequencing, we have a project on modeling the effects of COVID-19. We are working on data from connected devices or Internet of Things devices and mobile apps, and also on text analytics of medical records. So let's start the main panel right now. First, we have Dr. Ivan Koichev, who is a senior clinical researcher and consultant neuropsychiatrist at Oxford. He will give us an overview of the challenges related to the Alzheimer's disease in this day and age, no pun intended. As I mentioned, we are working on a risk calculator model with him right now. Ivan.
Thank you, Dan. Yeah, so as Dan was saying, I'm an academic psychiatrist working at Oxford. I've got a shared clinical and academic practice, and what I'm going to talk to you about is just an overview of Alzheimer's disease, as well as where machine learning would fit in all of this. So on the next slide is just a representation of the core pathology that we see in Alzheimer's disease. If we go to the next slide, yeah. So you've got amyloid plaques, first discovered by Alois Alzheimer's at the turn of the last century, and essentially these are intracellular deposits of a protein called amyloid. And then the second cardinal feature are these neurofibrillary tangles that accumulate within the neurons. And the interesting thing is that while amyloid spreads almost very homogeneously across the cortex, the neurofibrillary tangles tend to follow a very specific path, starting from the entorhinal cortex in the hippocampus, moving to inferior lateral temporal lobe, and then to the whole of the cortex, leading to this staging of Alzheimer's disease according to Brack. On the next slide is the main thing that I'd like to sort of convey, is that what we know now about Alzheimer's disease is that the process starts about 20 years before first symptoms. This is a graph from a study which followed people with genetic form of Alzheimer's disease, so people who have what's called an autosomal dominant mutation. Their parents develop Alzheimer's disease in their 50s. The children got tested and they found that they have this gene variation, which means that they will get Alzheimer's disease at roughly the same age. And then they followed them up with a variety of biomarkers from the early 20s. And what they found is that already at the age of 20, you start to see an accumulation of amyloid in the brain. About 10 years before first symptoms, you start to see this tau change. Remember, tau is the protein which goes into the neurofibrillary tangles and is the more toxic protein of the two. And then about 5 to 10 years before first symptoms, you start to get changes in the way that the nerve cells themselves work. So their brain glucose uptake, as well as changes in the actual structure using imaging. On the next slide is the thing that we now know about Alzheimer's disease, and that is that about 30% of all cases are due to preventable risk factors. So we no longer think about Alzheimer's as something that just happens to you as you get old. We now know that about a third of all cases are due to preventable risk factors, and that is super important because it means that there is a real advantage to actually diagnosing Alzheimer's as early as possible, or to identifying people who are at risk as early as possible, because there's a lot you can do to actually reduce the risk of Alzheimer's even without thinking about disease modifying treatments. On the next slide is the other main development in Alzheimer's disease, in that because we know so much about its biomarker profile, and we can use biomarkers to detect the presence of a variety of pathophysiologies in the living brain, we can now diagnose people biologically. What that means is that we're moving from a paradigm where we diagnose Alzheimer's on the basis of the history plus cognition. So at the moment, Alzheimer's is diagnosed as a dementia syndrome that is progressive in nature, and it affects more than 2 cognitive domains. And we're now thinking about Alzheimer's disease as a biological entity, where we look at whether they have elevated amyloid in the brain, that's the A marker. We look at whether they have amyloid tau in the brain, that's the T marker. And then we look at whether there's evidence of neurodegeneration, that's N. And so we're able to come up with this with these profiles, ATN profiles, where the browns that you see on the right are Alzheimer's pathology, and the blue are dementia or neurodegenerative processes, which are non-Alzheimer's in nature. On the next slide, I'm going to talk about cognition, how we deal with cognition at the moment. And on the next slide is a representation of how we do that at the moment in the clinic. So we mostly use pen and paper tests like these, where we go through a variety of tasks done on paper, and we assess people's ability to remember information, to deal with mental tasks. And on the next slide is where things are heading in the future, which is apps like these, for example, that we developed at Oxford, that leverage the now unavailable opportunity to test people in their own environment. This, for example, is what's called a paired associates learning task, where people are shown images and then they have to associate them with a particular direction. And then the great advantage of smartphones is that obviously you are not bound by the time people spend in clinic, but you can actually test recall of this information 3, 5, 6, 7, 8 days later. And so we're now talking about half-life of memory. So we all have a time point at which we've forgotten 50% of what we've recently learned. And we know that this half-life of the memory is what starts to shift first in preclinical Alzheimer's disease, so well before what we see in patients. On the next slide is where we will have even more data streaming in from digital devices. So, for example, moving beyond proactive testing, where people have to do these sort of targeted cognitive tasks, and thinking about how do people interact with digital technology, how long does it take them to find words when they think, how long, what is the quality of their speech when they speak on the phone, and also gathering data from what the unmentioned sort of the internet of things. So all of this distributed technology that we have in the house. In the next slide, I'm going to make an overview of if we could skip that slide and go to the next one. go to the next slide. Yep, next slide. Thank you. This is going to be an overview of the technology that we currently have to look at the other cardinal features of Alzheimer's disease. So amyloid scans are a positron emission tomography type of head scan, where you get tracers that bind specifically to the amyloid plaques. And then you can define whether somebody has a high enough level to class them as positive, amyloid positive, or whether they have normal levels of amyloid. And the significance of this, we know that people who are amyloid positive, they tend to have a significantly higher risk of developing Alzheimer's disease. This is one study which shows the cognitive decline of people who are either amyloid positive or amyloid negative. The amyloid positive are the red lines, and you can see there's a very clear separation within 18 months in terms of loss of cognitive ability as time goes on in between the amyloid positive, amyloid negative. On the next slide is the second main test that is currently being used, which is the tau PET scan. So this is again a compound which targets specifically the tau protein in the brain. And what researchers have found is that these tau proteins bind to tau as you would expect based on the clinical picture. So this, for example, are different subtypes of Alzheimer's disease, and the sort of the warmer colors is where the tau protein is. And so in a typical Alzheimer's disease, you would expect the pathology to be happening in the medial temporal lobe. So that's panel C, and that's what you see when you put patients like these through the scanner. You see this temporal lobe glowing. For example, if you look at posterior cortical atrophy, which affects the visual system, again using this scanner, this type of test, you can see that the tau is being picked up in the occipital lobe. That's panel A. On the next slide, we have the future of where the tau technology is heading, which is these blood biomarkers. So the main one is phosphorylated tau in blood, which has been shown to quite neatly delineate Alzheimer's disease from non-Alzheimer's disease pathology just by using a simple blood test. And then finally, we'll talk about on the next slide about neurodegeneration, which is the third cardinal feature. So currently we use imaging, where we look at the level of atrophy in the brain, and then specifically in the medial temporal lobe, and we class this as either Alzheimer's type pathology or not. But on the next slide is the future, which is again using a blood test, this time using an assay called looking for a protein called neurofilament light, which again has been shown to be sensitive to neurodegeneration. And then on the next slide is where machine learning fits into all of these. As Dejan said, a lot of these tests are quite expensive, and they involve radiation, and they're logistically challenging. So there's a piece of work around using data that's available on people to predict who it would be that is into one of these preclinical forms of Alzheimer's disease. And on the right is a paper we published showing a need separation when we use variables like age and sex and genetics. But we're currently working with Dejan's team to fashion this into a machine learning powered algorithm that can actually predict things on an individual level. Machine learning obviously has a role to play in the analysis of the huge data sets that are streaming through the digital devices, and we can always talk about that. And on the final slide, my thanks to you, as well as some details of a study we're running, where we're really trying to get to grips with what happens in preclinical Alzheimer's disease, testing the various biomarker profiles of people who are amyloid positive. That's what I had for you today. I'm very open to have a question and answer bit.
Thank you, Ivan. Yes, let's change the schedule a little bit, because I was thinking of asking questions to everybody after the three presentations, but I know that you have another call on the top of the hour. So let me ask you a question that I'm also gonna ask the other panelists. From your point of view, I mean, you mentioned several things that are coming in the future, but what do you think that would be the next big thing or the next big things that would make the most difference, say, in a midterm, like 5 years or so?
Well, there's two main things on the horizon. One is the other things that I mentioned. So the blood biomarkers are making a real splash at the moment because they turns out they're very sensitive to the underlying pathology. They can A, detect dementia, but also B, they can give you some idea about what the underlying cause of it is. So the etiology, whether it's an Alzheimer's type disease or whether it's non-Alzheimer's disease. So we're currently in the process of setting blood biomarkers in the actual clinical services, and that will really come to the fore over the next 5 years. And then obviously the second main development is the approval of the first disease-modifying treatment in Alzheimer's disease earlier in the year by the FDA. And we've seen a renewed interest into the field by a lot of companies that had previously pulled out of neuroscience and Alzheimer's in particular. So while the first drug itself, lecanemab, hasn't fared so well, it has definitely opened up the possibilities of an Alzheimer's disease modifying treatment gaining approval and being prescribed to patients. So to me, these are the two main things.
Okay, thank you so much for the thoughtful insight. And the moment you're going to see that we're kind of following a geographical order. We started on the West Coast in the US, then we went to the UK, and now we have Alexander Schliep, who is an associate professor in the Department of Computer Science and Engineering. And this is some kind of strange department that is between our partners at Chalmers and the University of Gothenburg. Alexander is on the University of Gothenburg side, but as I mentioned earlier, we are not holding this against him. Today, he's going to tell us about machine learning methods for antisense oligonucleotides and the applications to drug discovery. And also, he would mention a topic that is important to us about a mobility grant program that exists in Sweden. And this is certainly something that we would like to have in Bulgaria. So, Alexander, please.
Yeah, thank you very much for the invitation and the kind introduction. I still can't see my slides. They probably come up. There we go. Next slide, please. Okay, so the first part, as Diane pointed out, will be about science, and namely about design of antisense oligonucleotide drugs. And before I forget it at the end, I thought I'd thank my collaborators, Frederick Carlson at the ASO team at AstraZeneca, Shireen and Deborah Barotta at Chalmers and INRIA, who helped with the privacy bit, and then for the future work, Per Matsen at GU and Rula Zain at the Karolinska Institute. Next slide, please. So what are antisense oligonucleotide drugs? And as we all know, despite enormous efforts, pharmaceutical companies have not been always successful in identifying drugs for diseases like neurodegenerative diseases like Alzheimer's or Duchenne's. And these antisense oligonucleotide drugs are a new modality which have great potential to serve these unmet needs. The main difference is that classically drugs often interact with proteins, and drugs are based on small molecules. So you have small chemical molecules interacting with the proteins who do the signaling and are sort of the large formative building blocks of life at the molecular level. ASOs have a different modality in the sense that they prevent proteins from being produced. And what happens is that these antisense oligonucleotides, which are just short DNA molecules, DNA oligonucleotides, they bind to the messenger RNA, which is the part of the gene transcribed, and lead to cleavage and degradation, and thus prevent translation to a protein. Next slide, please. The And so instead of having to deal with the proteins, you prevent the proteins from being produced. There are two very important aspects to drug safety and to drug efficacy. For safety of a drug, of an ASO drug, antisense oligonucleotide drug, you want that these oligonucleotides bind to few or no off-target mRNAs. I mean, at every given point in time, up to 10,000 genes might be expressed in the cell, so messenger RNA for up to 10,000 of the roughly 20,000 genes in the human cell will be produced. And for F, you want that your oligonucleotides bind to the majority or preferably all of the target mRNAs. So you're targeting one specific gene, and you want to make sure that all the mRNA is degraded. And just to give you an idea about the scale of the problem, there are about 613 megabases in the human transcriptome and an in silico screen. So if you have a candidate ASO and you want to find out, does it bind somewhere where it shouldn't, and does it bind very well where it should, you have to screen against the 613 megabases. While there are, and for this is the binding energy. So Gibbs energy is the main determining factor. There are very good biophysical models available, but it's computationally infeasible to sort of screen that against the whole transcriptome. And approximate string matching, which is sort of the classical bioinformatics approach, sequence alignment, is a bad predictor for binding energies. Next slide, please. It is possible on the left to use a variation of these alignment algorithms to use a dynamic programming approach with some clever algorithms to speed that up. And we did this about 10 years ago for the same general problem, predicting hybridization to on and off targets in a different context of designing DNA microarrays, but I'm not going to talk about that in detail today. What we did, what we're currently doing is we try to solve that with a machine learning approach. And you can turn this problem to predicting the hybridization energy by binarizing that and saying, I want to know if an ASO candidate hybridizes to an off-target site. And I can just say if the binding energy between the antisense oligonucleotide and the off-target is less than the binding energy of the ASO and the intended target plus some threshold tau, then it hybridizes. If it's larger than that, it does not hybridize. So you, instead of computing a binding energy, you get a classification. And with that, of course, you can unleash the very large instruments or set of instruments for machine learning, which have been used for all kinds of interesting applications very successfully. And of course, features is always one important aspect of how to aspect of formulating things as machine learning problems. And what we're using here is we look at on one hand the ASO, on the other hand the intended binding site, and we want to classify how well they align by using descriptors of this alignment, which are easy to compute. So we don't compute an alignment, we just use descriptors like shared k-mers, which surface the features for the classification problem. And then we, as I said, we can use, for example, SVMs for this binary classification. Next slide, please. There's one interesting aspect to that. There are a lot of pharmaceutical companies. I mean, all the larger players are working on these antisense oligonucleotides. I mean, it's a very hot topic because it has shown promise for diseases which previously could not be targeted with small molecules. And so you're in a setting where every pharmaceutical company has their own little data set where they, test individual candidates with respect to their hybridization property, their efficacy, and their safety. And clearly, as there is commercial interest, a lot of this data is not published. And there is a way to actually use that and get classifier which is trained on all the data but doesn't require data sharing. So we actually used Federated Learning, and this is work with Shirin and Deba, which was published in a PharmML workshop this year at ECML PKDD. So we used federated learning. So we trained SVMs with ADMM. So we, in each site which contained a subset of the data, think of it as different universities or different pharmaceutical companies. We trained one individual classifier trained with all the data without actually requiring the sharing of the data. So there's no central node which holds all the data. Everybody keeps their data to themselves and trains them. And this federated learning per se is, of course, not privacy preserving, but you can do this in a setting, in differentially private setting. As usual, there is a trade-off between performance and privacy risk. As you can see on the bottom right panel, for the non-private performance in green, the dashed green line, we could match that at lower privacy levels. And so the sweet spot is roughly between 10 to the -1 and 10 to the 0. And so having reasonable amounts of privacies, you just have to pay for that with a small gap of accuracy. That, of course, was just a proof of concepts. We're continuing to work on that with AstraZeneca to scale that up. Astra has, in other settings, AI for small molecule drugs actually employed this privacy-preserving machine learning with great success. Next slide, please. And what we're planning, just to give you an idea, is now something that is not just the pharmaceutical companies for regulation reasons are mostly focusing on a few type of chemical modifications to these oligonucleotides. So they're not naturally occurring oligonucleotides, rather they have modifications. The most popular ones are linked nucleic acids, which change the backbone structure a bit. And there's a large sort of world out of there of things you can do to these oligonucleotides. And together with Per Madsen at GU and Rula Zain at KI, we just got funding for DDLS WASP grant. We're going to explore machine learning methods for predicting thermodynamics for novel modifications. And the idea is to use this active learning to identify most informative experiments, transfer learning because there is an abundance of naturally occurring thermodynamics data, a bit less for LNA, and we're interested in situations where there's very little data available. And we also want to play off simulations, classical chemical simulations, MD simulations, and machine learning to save simulations by using machine learning. On the other hand, provide extra data for machine learning with expensive simulations. But that is future work. We will be starting to hire and start working on this next April. I think this concludes the scientific part of the talk. Next slide, please. And next slide is, thank you. I'll talk a little bit about the strategic mobility grant Diane mentioned. So Sweden is quite particular in the sense that there is no classical sabbaticals for university faculty, but the Foundation for Strategic Research, SSF, has what's called strategic mobility grants, and that allows exchange of experienced researchers, and it's quite open. You can go from academia or research institutes to industry, government, or hospitals. It pays the full salary for ranges from 4 to 12 months. In my case, they paid 80% of my time in 2021 to spend at AstraZeneca, and I had an Astra badge, laptop, on-site access, etc., and very open discussions. And it was an absolute boon to get sort of new impulses for my research. On the other hand, to also carry, some aspects of, the state of the art of research into the sort of production work at AstraZeneca. And as you can see from the two plots, that sort of shows in black the total, in gray the number of exchanges from the university to industry, but also there is this very active exchange from industry to university, and there are a little bit less of the others. And at the bottom, you can sort of see the large players, ABB, of course, machines, AstraZeneca, Ericsson, Saab, Sandvik is mining, and Volvo cars. So there is a across all sort of industries, roughly according to, I think, the importance to the Swedish economy. And I think that's an absolutely fabulous instrument which very nicely augments the sort of education we give our students, preparing them for the industry. And on the other hand, making sure that we can educate our students well enough by having a good feeling for what industry really requires. And with that, next slide, I want to thank you and close. Thank you very much for the opportunity to present, and I'm happy to answer questions.
Thank you, Alexander. If we had more time, I would have asked you in more detail about that mobility program. I mean, as I mentioned, this is something that we really need badly in Bulgaria. As somebody who is and has been on both the academic side and the industry side, there is perhaps larger disconnect here, and it seems that in Sweden you have good traditions and a system that works nicely in place in order to have a collaboration between academia and industry. However, as I promised, my question is going to be about the future. You mentioned a couple of things that are coming next, but out of them all, is there something else that you think that in kind of mid-run would bring the most advancements of the technologies? What would that be?
I mean, I think there's one sort of elephant in the room people like to ignore, and that's just the sheer amount of human genetic variation. I mean, if we look at drug design, for example, it's very clear that the best data is available in the US, that African Americans are very underserved, both by sort of clinical care, diagnostics, but also by treatments. And the reason for that is underrepresentation. And if you look at sequencing efforts, right, I mean, there's an enormous amount of DNA, and not just structural variants, but really novel DNA, which we are blind to because we just don't sequence deeply enough. So I think that these large sequencing efforts and switching from individual reference genomes to pangenomes, to references which present the full variability of human variation, will have an enormous effect. I mean, to make this very concrete, one of the things we have on the radar I haven't talked about is we are basing the drugs efficacy and safety is based on how well does a part actually hybridize to a gene, right? And if you have variation in there, you will have an enormous spread. And so if you just work with one reference genome, you can design very nice drugs which work with the white European male who most likely contributed to the reference, but you'll end up with drugs which might not be safe or might not work as well for other populations. And given the enormous range of migration and we see in Europe, right, this is not just a problem elsewhere, this is a problem everywhere in the world. And I think that is sort of the biggest challenge for the next 5 to 10 years, I see.
Okay, thank you. And indeed, combining different data, in this case, different genomes, is a significant challenge. And if you remember, I mentioned that one of our projects is of an omics type, which combines different types of data. And this is not a trivial problem to solve. Our next speaker is Todor Klimov. He's the head of life sciences and healthcare solutions at OntoText, and he's going to tell us about multimodal knowledge graphs and how, among other things, they can be used to incorporate different types of data. Todor, please.
Thank you for the invitation and also for the introduction. As Dejan already said, I have the opportunity to explain what are multimodal knowledge graphs and how they can benefit to develop really intelligent decision support systems, both care and pharma. We'll go through a very brief introduction of the company and what we do and why we believe in knowledge graphs. Then we will explain our recent accomplishments in several commercial and research projects which are based on knowledge graphs. And then we'll point out some interesting development of application of knowledge graphs actually in Alzheimer's disease. Yeah. So Ontotext is a Bulgarian company which is actually a leader in the knowledge graph development. And as such, we have created our core product, which is an RDF triple store, which we position in the core of all our solutions that we develop for various domains. It allows us to integrate structured data, the metadata about these data sets, unstructured content, to extract and to formalize the knowledge in these data sets and these data sources, to build a really huge holistic knowledge graph that covers different aspects of one single research domain, to align it with explicit knowledge expressed in industry-specific ontologies and terminologies, to apply various learning algorithms on top of to infer or to generate new data on top of the knowledge graph, to augment the knowledge graph with this newly generated data, and then to bring really deep insights over a research problem. So why knowledge graphs? I will not go in very technical details, but what we do with the knowledge graphs is actually we decompose the knowledge that we like to integrate into single facts. Each single fact is expressed by either a node from the knowledge graph, it could be a G, and another object to which it is actually related. And the relation is expressed as a predicate having the actual semantics of the relation, which can be both interpreted by humans and also systems, which is also very important. Within this process, we actually aim to align all both the objects, the nodes in the graph, but also the predicates, the relations between these objects, to ontologies. Why? Because we can use the formalized in the ontology logic to infer new information based on the actual instance data that we populate in the knowledge graph. Then once we have the knowledge graph, we can easily do many things. We can use it just as a normal database fitting, but which is more, we can benefit from the actual graph. So we can traverse the graph to find interesting relations, new hypotheses, but for this to be accomplished, we need really a very comprehensive, very rich knowledge graph, combining data from many different sources, many different modalities of data, in order to give us the holistic view over a simple problem that we would like to solve. And as it is illustrated in the diagram on the right side, we can really load various types of modalities, starting with proteomics, genomics data, clinical profiles of patients. We can normalize these data sets to reference some recent terminologies, and we can harmonize the data so that once we load all the different modalities into one single RDF triple store like the product we have, it will automatically build a knowledge graph that then can be queried and explored and mined for interesting relations. And then you can apply also various types of machine learning, deep learning algorithms, graph embeddings especially, that can be used to infer new information out of the high quality data that we have in the knowledge graph. I'll focus on two reference projects in which we have built really very big knowledge graphs. The first project, we developed a knowledge graph for a top 5 pharma company, and it was aiming to really enable preclinical research teams to mine for various of relations between different types like genes, molecular interactions, pathways, associations with genes and diseases, associations between biological processes and certain disease conditions, and to infer really new information that can be used to identify newly potential biomarker targets that can be used for their drug development. So in general, what we did was to integrate various public data sources covering the different modalities and to normalize them to corresponding images, then to integrate all the data in a single repository, resulting into more than 1.3 billion facts, significant amount of which was also inferred data that was not already presented in the source data. And we have tested at least 5 different use cases for finding, for example, different type of associations between the data. The other project is a research project funded by the European programs. And it's actually, the project is called ExaMode. It's actually, the objective of the project is to build a multimodal knowledge graph, integrating data from the clinical systems at the hospital, and also automatic analysis of the images, histopathology images, in order to build a holistic knowledge graph that will facilitate the decision making in the process of diagnosis of different cancer-related conditions. We're focusing on 4 diseases, one of which is colon cancer. The current process is taking the sample from the patient, preparing the slides, then covering the tissue samples, then digitalizing or scanning the actual slides and transforming them into high quality images that then load a centralized repository of whole slide images, which then are analyzed with deep learning algorithms to identify the different formations that can lead the actual histopathologists to the conclusion that this patient is can be diagnosed with this particular condition or it's not. In addition, we also use the patient information from the laboratory information management system coupled with the hospital information management system, which also provide valuable information about the patient profile, demographic data, current diagnosis, other site information that can be used in order to facilitate the process of diagnosis. We use a couple of public data sources, ontologies, in order to prepare the ontology that will be used to build the model, which then we need to populate with both data coming from the analysis of the images, but also extracted from the medical record. And we have created a text-based encoding such medical texts based on SNOMED CT codes. We take all the information which is available in the clinical record of this particular patient, and we finally, after we process the data with the pipeline, we assign diagnostic codes which are aligned with the SNOMED CT terminology. And as a result from the project, we actually developed a solution prototype, which is a software platform for building holistic solutions from multimodal data. In this case, it's histopathology data, which help the medical to make make more informed decisions based on various type of data, to prioritize the cases which are based on severity or likelihood of a specific should be processed first, and really to use one of the powerful features of the knowledge graphs, which is based on graph embedding to calculate similarity of patients in this case, which are single nodes in the knowledge graph. We can really very easy compare how a case report to which other case report knowledge graph corresponds, or which are the most similar cases, in order to check what was the diagnosis there, what was the treatment, and so on. As the knowledge graph actually represents the main ontologies and the terminologies, this is actually the referential knowledge that we use to normalize the data coming from the clinical records. This diagram actually represents 3 different clinical cases with the information already normalized to the referential ontologies about the diagnosis, the morphology, the tissue sample, and so on. So then it can be really easy queried, compared, because all they refer to one and the same objects in the knowledge graph. It's not actually textual data, but already normalized and harmonized data. And then, as I said, it can be easily developed a model, statistical model, that you take into consideration specific categories of information like diagnosis, procedures, dysplasia in this case, tomography, topologies also, and some demographic data that then can be used for calculation of the similarities across different cases. Now, because we're interested in Alzheimer's disease, I wanted just to point out to 2 interesting research activities which apply the same approach, building knowledge graphs to solve specific problems in the treatment of the disease, whether it's to understand much more in detail what are the molecular mechanisms behind this particular disease, or what are the potential treatment pathways that needs to be followed. One is the neuro RDF, which starts with the very similar approach that we usually apply, with the initial data collection, using data sources, structured data, to mining literature, and also combining with gene expression data. Then some absolutely necessary data quality and curation and pre-processing step in order to validate the source data that then can be used to really build a high quality knowledge graph based on the RDF standards and models. Then on top of this knowledge graph, then can be developed different data mining and analysis methods that can point out specific findings in the knowledge graph build. This project actually is most focused on molecular intelligence. So the diagram on the right side, it depicts the complex interaction networks between the proteins related to this particular disease. The other project provides much more systematic and very well described overall approach how such a system can be designed and implemented. Started with a really high-level ontology upper model that can be used actually to model any particular disease condition, not only Alzheimer, identifying the main entity classes and the most important relations between them. Then taking this source model and adapting it for the particular needs of this disease, Alzheimer's disease. And on the right side, actually populating the model with instance data collected from all the available public sources, and then using this data to consider each particular case report or patient record to what extent aligns with the particular model. So if you're interested, you can check the research articles which I listed below each for further information. I would like to acknowledge the ExaMol project, which was used to develop histographer solution and was referred in these slides. And with that, I would like to thank you for the attention, and I'm open for questions.
Thank you, Todor. I should say that your technology is perhaps the thing that I'm the least familiar out of everything that was presented today. So, same question to you as to the other of our panelists. What is the thing that would bring the most advancement for the work of your team and your company in the next several years?
I will try to formulate it a little bit different because, well, in the last 15 years, which I spent in the company working with this technology and solving different problems in the life science and healthcare domain, we usually do a reverse engineering of some data generated with a conventional approach, whether it will be a relational database that describes some entities and the relations between them, or a research article that expresses some specific research and findings. We always try with our technology to extract the knowledge out of structured and unstructured data sources and to formalize it into useologies and semantic models. In order we as a community and as researchers to benefit from the semantic technologies, we need to think of completely different way, not to extract the knowledge and to formalize it, but once we generate the knowledge, to be semantically ready, meaning that if the author himself doesn't depict the precise context of the information he is creating, then no one else later on down the stream of the processing of this data will be able to capture the context he's putting in his data as precise as he. So we need to complete the attitude how we generate data and how we maintain it. The semantic modelization happen when you
so
create
it's
the
not
data.
a quantitative difference, but I would say qualitative difference in the way you approach your thinking about the data. Okay, thank you, Todor. Also, I would like to thank Ivan and Alexander for taking part in our panel today. Now we have run out of time. According to the agenda, we were supposed to show brief presentations of our younger researchers who are doing work in the digital health domain. However, there is not enough time to do this right now. So these presentations would be shown during the time that was originally designated for the lunch break. This is 1:30 p.m. to 2 p.m. Bulgarian time, or 12:30 to 1 Central European time. However, I would use the remaining couple of minutes that I have until the beginning of the next session, that is City Digital Twins, to very briefly present these researchers and their work. First, we will have Sophia Wazarova, who works on ontologies. That's an important work because often we have all sorts of data, all sorts of parameters and features that are in the data, and we need a good understanding of what is in our data, what are the relationships between the different data sources and data points that we have. And practice shows that without it, we can be really lost in our understanding and even draw wrong conclusions by having a good and real data. Next, we would have Denitsa Grigorova, who is working on a topic that I mentioned earlier, imputing missing data. As I said, especially in clinical trials, there is often missing data. That could be people missing their appointments or dropping from studies, or certain types of tests not being done for some people, depending on which facility they go to. And it would be not a good idea to just drop these observations and not use them. And for that reason, we are working on imputing missing data, which is filling in the missing information with reasonable values. Also, I mentioned earlier before that in many cases, even if we have good data, there is not enough. That sometimes is related also to privacy issues. And for that reason, a good approach would be to generate synthetic data. And one of our colleagues, Felicia Shakova, has been working on that topic for the past couple of years. And finally, Evgeny Marinov will show us some of the machine learning methods and technologies that he's applying to the pre-scoring calculator for Alzheimer's disease. And again, the presentation of the young researchers that was supposed to be broadcasted right now, it's a pre-recorded presentation, would be shown during the lunch break at 1:30 Bulgarian time or 12:30 Central European time. Thank you.
Hello everyone, my name is Sophia Wazarova and I'm junior researcher at the GATE Institute. Today I'm going to shortly talk about ontologies in general and the ontology I'm building for the Cognition project. So by the end of this talk, I hope that I would have answered the following three questions. What is an ontology? Why do we need ontologies? And how do we apply ontologies here at GATE? So when we're talking about ontologies, the very rough definition of it would be a toolbox that is allowing us to model the world around us or parts of it. But what really makes ontologies special is the fact that they are taking this classical taxonomy idea and they're expanding it a little bit with definitions for all the classes. And then they're taking this very nice but human readable structure and they're translating it into a machine readable definitions. And by doing so, ontologies support automated reasoning and they also support knowledge discovery. So as an outcome of this, we can say that we are using ontologies whenever we have very complex domains that we need to describe and we have a lot of people involved and we want to make sure that all of these people have access to the same information and understand it the same way. Whenever we are integrating data from multiple sources or we would like to enable automated reasoning and knowledge discovery. So the Cognition project is a project that's dealing with modeling patients with cognitive disorders and ultimately is aiming at using machine learning for finding out early predictors of such disorders or just predicting the outcome for patients in a more effective way. So how did we decide that we need ontology for this project? Well, for one, we have a lot of people involved and a lot of them have different backgrounds. We have physicians and we have software engineers. And whenever these people are talking to each other, we would like to make sure that they mean the same thing and they're using the same vocabulary. Apart from that, medical data is very complex, and for anyone who's not part of the medical field, this could be quite confusing, and ontologies can be very helpful here. And last but not least, this project will feature data integration from multiple sources, very complex data, and also from sources that have different types of data. So ontologies can be very helpful here as well. The structure that our ontology is following is that it's taking this subject, which might be a patient or it might be a healthy subject, and the subject is going through a number of medical tests, and the results of them are actually clinical findings, which are kind of put together, forming this clinical picture, which is then evaluated, and the outcome of this whole process is the current diagnosis of this patient. And at this current time, we are focusing on Alzheimer's disease, so we are taking this general structure and we are plugging in data that's relevant to diagnosing Alzheimer's disease. So practically right now we have 108 classes, and this is just a partial image of the class taxonomy that we are currently using. But building an ontology is actually just the first step of many ahead for Cognition, and right now we are at the point where we're going to begin this big data integration, by the end of which we are hoping to get large enough database and on which we can then focus on data analysis and efficient visualizations. That's it from me. Thanks for listening and enjoy the rest of our event.
Hello, everybody. Thank you for being here and listening to my presentation. My name is Denitsa Grigorova, and I have a PhD in statistics. Currently, I hold an experienced researcher position at the Digital Health Unit within GATE. I joined the team in September last year. Back then, I started working on the data from the Alzheimer's Disease Neuroimaging Initiative, abbreviated as ADNI, which is a longitudinal multi-center study designed to develop clinical, imaging, genetic, and biochemical biomarkers for the early detection and tracking of Alzheimer's disease. Longitudinal studies are studies that follow up subjects over time, and the data consist of repeated measurements on the same individuals. My main research topic is missing data with application in Alzheimer's disease. My most current project is to adequately model the results from one cognitive test called Mini Mental State Examination, or shortly abbreviated MMSE. It includes 30 questions on orientation, attention, memory, language, and visual spatial skills. The score is the number of correct answers, which means an integer between 0 and 30. This test is commonly used in medicine to screen for dementia. Advantages of the MMSE include requiring no specialized equipment or training for administration, and has both validity and reliability for the diagnosis and longitudinal assessment of Alzheimer's disease. Here I present the histogram of the MMSE score at baseline on ADNI data. From it, we can clearly see the ceiling effect. People with normal cognition very easily get the maximum number of points, which is one of the challenges of modeling this type of data. This is a spaghetti plot of MMSE score on 10 individuals over time, over 4 time points, baseline, 6 months, 12 months, and 2 years after the baseline measurement. Some of the subjects are with normal cognition in red, with Alzheimer's disease in black, and late mild cognitive impairment in green. We can see that some individuals dropped out of the study. Missing data are a very common problem in longitudinal studies. Very often individuals drop out or have intermittent missing observations. If not properly accounting for the missingness, the statistical analysis may lead to biased results. Now, let me tell you some words about the statistical definitions of missing data that were introduced by Rubin in 1976 and now are widely used. I will cite the examples from the paper by Scott and Bartlett for the illustration of these definitions without giving technical details. Okay. There are 3 missing data mechanisms used in the literature. Missing completely at random, for instance, is a failure to attend for a scheduled psychometric assessment in an Alzheimer's disease trial because of a taxi breakdown. It is as likely to affect one individual as any other, independent of the data already collected or the data that were failed to be collected. Missing at random is, for example, a failure to attend for a follow-up psychometric assessment that was related to possibly unmeasured psychometric performance, but this association could be fully explained by dementia severity at baseline. Then any missing psychometric follow-up is missing at random. Missing not at random is a subsequent missing data when the patients dropping out of the Alzheimer's disease trial were those undergoing rapid cognitive decline following their last follow-up visit, and this could not be predicted from the data already collected in the trial. While one can distinguish data missing completely at random from missing at random or missing not at random, using relatively simple statistical techniques, it is impossible to distinguish the latter 2 categories by analyzing observed data. In my current work, I explore and practice data analysis techniques that can produce unbiased results under different missing data scenarios. For example, Now we apply multiple imputation technique, that is one of the statistical procedures for coping with missing data problem, on another Alzheimer's disease dataset. It is very important to properly account for the missing data problem to obtain reliable statistical results. Thank you very much for your attention.
Hello, my name is Felicia Shakova. I have a PhD in molecular biology from St. John's University, USA, and a master's in bio and medical informatics from St. Clement Ochridsky, Sofia. I'm an experienced researcher at Gate Institute working in the domain of digital health. I work on multiple projects involving data analysis and machine learning techniques for biomedicine. My pilot project is concerning the application of classification methods on synthetic RNA sequencing data. Rna-Seq is a modern and ubiquitous genomic tool that has greatly enhanced the understanding of the function of individual genes and gene networks. Rna-Seq experiments have led to a number of discoveries in medicine over the recent years. This includes the stratification of acute myeloid leukemia patients into cohorts requiring different treatments, an example of personalized medicine. Also, the finding of key factors with important role in the pathogenesis of autism spectrum disorders. The experiment detects and quantifies multiple messenger RNA molecules, product of gene expression, described by the term transcriptome. The Rna-Seq data is obtained by a standard workflow that includes a laboratory and a computational part. The computational part is extensive. It includes read alignment to a reference genome, quantifying reads that overlap a certain gene, representing the measurement of expression level. This is followed by filtering and normalizing and statistical analysis of significant changes in the expression levels of individual genes between sample groups. This is the most common Rna-Seq application, the so-called analysis of differential gene expression, which is now considered a routine research tool. A well-known problem in the analysis of Rna-Seq data is its high dimensionality. The expression of tens of thousands of genes or features is being measured, while the number of tested patients or samples is often restricted to a few hundred or less. This is due to hypersample cost, rarity of phenotype, or privacy restrictions. An example of the presented challenges is overfitting, when the classifier separates the sample data at hand well, but does poorly on future data, leading to unreliable prediction. When comparing machine learning methodology for Rna-Seq data analysis, it is also necessary to measure the performance accuracy. Usually, this is not possible to do on real data sets due to unknown true gene expression levels and true differential expression state of each gene. With synthetic data, we can introduce changes in a controlled fashion and choose the optimal machine learning classification model with reliable error estimation. In a collaboration with Associated Professor Ivan Ivanov at Texas A&M University, we have conducted a study that examines the influence of different model parameters for the generation of synthetic Rna-Seq data on several classifiers. These are linear discriminant analysis, LDA, k-nearest neighbors, KNN, support vector machines, SVM, and artificial neural networks, ANN. Here are our results. The panels A and B confirm the expectation that the increase in dispersion would lead to an increase in error rate for all classifiers. This is based on the fact that there could be an overlap between the distributions of the two classes when their variances increase, which makes them not as readily distinguishable. SVM is the most accurate classifier in the majority of the considered simulated data scenarios. However, the simplest classification rule, LGA, has similar or in some cases even better accuracy when the number of training samples is small. This is the result for the true positive rate metric, the classifier with the highest mean and lowest variance overall are SVM and KNN. LGA shows a high mean in the case of 50 training samples. LGA shows the greatest variance compared to the rest of the classification rules. This suggests that when only a small number of samples of real data is available, the standard scenario in biomedical research, LDA could be recommended as it is both simple and accurate classification goal. Thank you very much for your attention.
Hello, my name is Evgeny Marinov. I'm an experienced researcher at Gate Institute since September 2020. My main focus at Gate is the research and development of machine learning algorithms and exploratory data analysis. I'm currently working on a project related to Alzheimer's disease progression based on the so-called ATN framework. ATN stands for amyloid, tau, and neurodegeneration. Our current project is an extension of a research paper of colleagues from Oxford University. Predicting ATN status in pre-dementia individuals provides an important opportunity for targeted interventional studies. 1,010 other adults that are part of the study were allocated to one of the 5 ATN-defined biomarker categories. In the previous research, multinomial logistic regression tested risk factors to predict one of the 5 ATN subgroups. Some of the factors or features, including age, sex, education, APOE4, genetic variations, family history of dementia, cognitive function, vascular risk indices such as body mass index, high cholesterol, physical inactivity, ever small blood pressure, and some others are used as features for the machine learning algorithms. Based on the measures of amyloid beta, p-tau, and neurodegeneration, there are 5 target AD groups categorized in the ATN framework. These are the normalized AD biomarkers, including 56% from our data, Alzheimer's pathologic change, consisting of 21%, Alzheimer's disease 60.6%, and so on. In our research consists of the introduction of additional features based on information value and weight of evidence. Weight of evidence helps to transform a continuous independent variable into a set of groups or bins based on similarity of dependent variable distribution, that is number of events or number of non-events. And for continuous independent variables, we create bins or groups and then combine these categories with similar weight of evidence values. Some rules of the weight of evidence are that each category should be non-zero for both events and non-events. The weight of evidence should be monotonic, either growing or decreasing with the groupings. Missing values are binned separately. Another addition to the initial research consists of the application of more machine learning algorithms and methods applied for generalization of future or so-called unseen by the algorithm data. Some of the machine learning algorithms are tree-based methods like XGBoost, random forest, and there are also more standard ones such as support vector machines that we used in our research. Another methods from the machine learning domain we apply is the k-fold cross-validation. Cross-validation is a resampling method that uses different portions of the data to test and train a model on different iterations. It is mainly used in settings where the goal is prediction and one wants to estimate how accurately a predictive model will perform in practice, or how well it will generalize for unknown by the algorithm data. For the validation of our machine learning methods, we apply the area under the curve, which is a measure of the ability of a classifier to distinguish between classes, and it is used as a summary of the ROC curve. The higher the ARC, the better the performance of the model at distinguishing between the positive and negative classes. An ROC curve plots the true positive rate versus the false positive rate at different classification threshold it's shown on this slide. And applying of these methods, we are able to improve the previous results by several percents, which is even more significant, although we use cross-validation. Thank you for your attention.